This video is about bone development, and bone development has several topics. For example, just generally, how do we go from an embryo made of 4 cells to an adult that has a skeleton? There are a lot of different steps in that process. One of them would be making a hyaline cartilage skeleton in the fetus. Another would be making some of that hyaline cartilage, transforming it into bone. And so we see a 12-week-old fetus here. And one thing to look at is the long bones. Where do they meet? I mean, they haven't fully elongated. In places like the carpals and the tarsals haven't formed at all yet. They're still hyaline cartilage. So how does that work? And then how do long bones get longer? How do these bones form? Because the bones of the skull form in a different way, and they have to form pretty early to protect that developing brain. So how do they form? So those are some of the things we're going to cover here. And then finally, how do we remodel and repair bone? The textbook, which I believe you can get for free through the library, has a lot of information about bone breaks and healing that we have to skip here. But it is fascinating stuff. And so I do urge you, remind me, and I will read all those emails I got late last night about the textbook, and if you can get it for free, go get it. Okay, right. Here we see an early embryo, or a week 7 embryo, that has only hyaline cartilage and some mesenchyme. Now, what is mesenchyme, and why do you remember that word? 3, 2, 1, pause. This is the tissue that gives rise to all connective tissues in an adult. So an embryo has mesenchyme, and the cells in that mesenchyme, they divide and have daughter cells. Their daughter cells divide and have daughter cells. Those cells divide and have daughter cells. And eventually some of those daughter cells become bone. Some of them become cartilage. Some of them become blood cells. Some of them become CT proper. So that's mesenchyme. And so bone is going to appear around week 8 and slowly replace everything else. There are 2 types of bone development, and in particular, I should clarify, there are 2 types of ossification. And ossification is the process of making bone from something that is not bone. The 2 types are intramembranous ossification, which is among or within membranes. This is how we get flat bones of the skull and part of the clavicle. The cranial bones mostly, like the parietal, for example, occipital, temporal, the ones that have those big flat plates, we get them in this way. And then every other bone, endochondral, endo is within, chondr, that has to do with cartilage. And that means transforming cartilage into bone. So these form directly from the mesenchyme in a membranous way, and these don't. These form from hyaline cartilage. Yeah, cool. So intramembranous is the simpler to envision, or perhaps it's more complicated, but the details are at the cellular and molecular level that is way beyond this class. So we won't go into how intramembranous ossification works, just understand this is what you get. A flat bone with compact bone in 2 layers and spongy bone in between. Part of the clavicle is made this way, but the bones of the cranium are made this way. And so these are formed directly from the mesenchyme. And it's an interesting process, but most of that process happens, again, at a level that we would not see using the tools we use here in anatomy. So in anatomy, we try to look at things you could see under a microscope, And intramembranous ossification, all the interesting stuff happens in how the cells get reprogrammed to change their behavior. And that's not what you can't see that with a microscope. So forget that. We will look at endochondral ossification because this is essential to how we grow. And this goes on all through childhood and adolescence to make us taller and longer and make our fingers longer, etc. This requires hyaline cartilage to get remodeled and break down before it can become bone. This slide I thought I had deleted. Sometimes I edit slides, and if I want to skip a topic, but you might be curious about it, I change the background on the slides to make them gray. I did that last semester. This semester I just deleted them. So I wish I had also deleted this slide, but I didn't. Okay, so what is an epiphyseal plate? Where have we seen those? 3, 2, 1, pause. An epiphyseal plate is the growth plate in a long bone that makes a long bone longer. And so it's between the diaphysis and one of the epiphyses. So how do they grow? How do they get longer? Well, this is a tricky conceptual thing, and I want you to bear with me, and then you'll have to work through it. What I'm going to show you is this ugly looking picture. This is a picture, I believe this is, under the microscope, of the growth plate or epiphyseal plate. And it's a lot of stuff you can't interpret yet, but what I want to draw your attention to is the 5 distinct areas. So this is one distinct area. This is one area that definitely stands out. This is an area of transition between things that look like this and things that look like this. This is distinct, and this is distinct. So what we're going to find is that as the epiphyseal plate grows, what it does is it keeps moving away from the diaphysis. It keeps growing towards the epiphysis and leaving bone behind it. So it keeps leaving new bone on the diaphysis to make the diaphysis longer. And it pushes the diaphysis away, little by little. As it does that, we can see a kind of a 5-step process happening. And those steps happen slowly, and it's slow enough that you can actually see different regions in the epiphyseal plate where cells are going through those 5 different steps. And so, blah, 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 5 steps, 5 steps. Yeah, and so the first step, the step up here would be cartilage that is the first thing, it's the front. And down here would be actual bone that has been laid down. So this would be healthy hyaline cartilage, and this would be bone, and this would all be cells that are either changing the shape of the epiphyseal plate or becoming bone. And so zones. So here it's been drawn out for you, and it's much easier to understand when it's drawn like this. What we have is these 5 zones. We have a resting zone, and this would be hyaline cartilage. And like all hyaline cartilage, the chondrocytes don't do much. They don't get much in the way of nutrients. But eventually they get a signal that says, hey, by the way, it's time to move this epiphyseal plate and make the diaphysis longer. And so what they do is they start dividing very quickly, and they line up so that their daughter cells are going to be on these 2 sides, and this one is going to have its daughter cells like that, and this is going to have its daughter cells like that. So they're not going to divide randomly, they're not dividing horizontally, they're all dividing vertically. So that happens very quickly, and you end up with these stacks of lacunae and chondrocytes. So these are the cells that have gotten that signal. These cells have not yet gotten the signal. So these are just the hyaline cartilage. These have been told it's time to become bone. But first, move the epiphyseal plate. So as these proliferate, they push in both directions, and that pushes the cartilage and the epiphysis away from the diaphysis and makes the bone longer. These cells were doing this earlier. They got the signal to divide earlier. So these have all already been pushed apart by this process, and they're done dividing. So what they do is they mature, and they get very big, and eventually they stop dividing. So the hypertrophic zone is where they begin to start calcifying, and they begin to stop really being cells. And then the calcification zone is where they die, and the matrix calcifies, and cartilage breaks down. And then the ossification zone, this is where blood vessels will invade, and osteoblasts and osteoclasts will get to work shaping this. So those 5 zones kind of show you the whole epiphyseal plate, from the leading edge, where there is hyaline cartilage, to the trailing edge, where it leaves bone behind it. And in each case, it's cells that have gotten a signal, and then they've divided, and then they go through hypertrophy, and then they die and ossify, and then they are converted to bone. So those are the steps in endochondral ossification. And here you see the that picture I showed you before. Here an artist has redrawn it to try to make it a little bit clearer and show you the zones. And all of this would be taking place in the epiphyseal plate of an adolescent or child. And you can see the epiphyseal plate because it's not bone, so it doesn't show up on an X-ray. So in this X-ray, it shows up as a space. And again, we have the resting zone. This would be hyaline cartilage up here. Proliferative zone, where the cells are rapidly dividing and trying to push, basically. And then as they get older, they get bigger, and they also push. And so this is where they really start to lengthen the zone and move the epiphysis away from the diaphysis. And then they die, and from below, the bone kind of invades and reorganizes. So that's endochondral ossification. I realize it's not a super intuitive process, but what you have to do is think of it in terms of those 5 zones as 5 steps. And one thing that can help is asking yourself, okay, these cells here, what are they going to do next? What did they do before? What did they look like before they looked like this? What will they look like after they look like this? Do that for all of these zones, and you will understand endochondral ossification. So then we get these big patterns of how the bones develop, and that's what this is showing us. So during childhood and adolescence, bones lengthen through endochondral ossification at the epiphyseal plate. And yeah, the epiphyseal plate maintains a constant thickness, right? So as it pushes towards the epiphysis, it lays down bone behind it. And so a new region becomes the epiphyseal plate. And then towards the end of adolescence, that slows down and the epiphyseal plates become thinner and thinner until eventually they're gone. Eventually, in adulthood, they become epiphyseal lines. Also, bones have to get wider and thicker, and that happens as osteoblasts and osteoclasts remodel the bone. So they work in the endosteum and periosteum, and they remodel the bone. Remember that there were two growth patterns. There's appositional and interstitial growth. Well, endochondral ossification is interstitial growth. It's from within a tissue. Appositional is growth on the outside of a tissue, like what you would find in the periosteum. And that's how these bones get thicker. So osteogenesis makes the bony skeleton, converts it to bone, thickens, repairs bone. And what I want to leave you with is some ideas about bone remodeling. So bone is a dynamic living tissue. That means that 500 milligrams, or half a gram, that's a visible amount, that would be a visible pile on your hand, leaves and enters the skeleton every day. So that calcium is cycled in and out between the bloodstream and the bone every day. Also, spongy bone is replaced completely, as is compact bone. Compact bone is replaced every 10 years. What does that mean for you? Well, bones will develop based on the stresses we put on them. So if you go running every day for 10 years, your bones are going to get stronger in a way that supports running. It may take 10 years, but after 10 years, your bones are going to be better for that purpose. If you want your bones to be strong, you can work out, use weight machines, and if you make a habit of that for 10 years, you will have a stronger skeleton by the end. And that will start much faster than 10 years. But after 10 years, you can completely transform your skeleton, or within certain limits. Let's see, what else? I'm going to skip all this. So 51 is the last slide. We're going to skip details of bone remodeling. You have to know what the different cells do. You have to know what an osteoclast does and what an osteoblast does. You should know that by now anyway. But we won't look at the patterns or anything like that. So I guess I should make all these gray, and they will not be on the exam. So we're done with bone development, and that's it for Lecture 5. Lecture 5, Part 1. This is a big lecture with a lot of information, but you can handle it. A lot of it is just memorizing diagrams, and you know how to do that. So get to it, and I will see you soon.